Welcome students. This is for the 6th semester students for Herbal Cosmetics, the chapter number 4 or unit number 4, lecture number 2. As I told, we have discussed about the questions and the general cosmetology. We have discussed about the natural cosmetics and examples. And now the second lecture for this unit in Herbal Drug Technology of Unit 4. Here in this part we are going to discuss about the herbal excipients and herbal formulations. They used to ask even for the 8th semester students for quality control and standardization of herbals and herbal products, the subject they used to ask. So it's necessary for the students should know about some details and some information about the herbal excipients. What is its difference? What is its advantages from the conventional or the synthetic excipients and additives which we are using in our pharmaceutical industry for various formulations? So it's necessary for the student should know as a pharmacy, B pharmacy or Pharm D. All the students, including diploma in pharmacy students also, they should know what type of natural excipients and what's its advantage and what's available with us can be used, and it will be helpful for the mankind. Herbal excipients, we know that we have studied, you have studied, you might have come across excipients, additives in formulations, in pharmaceutics, and you should know about herbal excipients. Excipients in pharmaceutical aid, you might have used the word pharmaceutical aid. A compound or an chemical or any substance which have been used for in the process or in the application of any type of manufacturing of formulations. We used to say it's used for pharmaceutical aid. Even excipients also pharmaceutical aid are the substance which are inert and has little or no therapeutic value. They are inert. Mostly they don't have any therapeutic value. They are, apart from the active ingredient, they are supportive material to give the strength and consistency, and to make the formula we are using these excipients, additives or pharmaceutical aids, but are essential in manufacture of various pharmaceutical dosage forms such as tablets, capsules, syrups and extra many. Even we know that many semi-solid preparations are also, it just we put etc. because lots of new novel formulations are available. Excipients are mixed with active ingredients. Repeatedly I am telling some words to make the students to listen to that. Active ingredients to make up the volume. Sometimes we used to use fillers, diluents to the potent drugs. So these all are things which it should come from your mind, and as well as it should be in your fingertip when you are writing an exam. When you are writing a question to be answered in the examination point of view, the student should know how many points what you studied in the lower classes and in your semesters should be always remembered when you study advanced courses. So to make up volume or to improve the stability, sometimes we used to use preservatives, and its stability and release of the drug all are controlled by the excipients, and mask the bitter taste. Sometimes it is bitter taste can be masked by means of adding some excipients, coating. This all comes in an excipients, or improve the appearance. It's very important for the herbal formulations. Sometimes you say when you are taking crude form of herbal formulations, tablets or capsules or powders or granules, when you take it will be having a different color which may not be acceptable by the patient. So you should improve the appearance, not only by means of appearance by color, even the odor, even the flavor, even the texture can be altered by means of using some excipients. Other characters of the dosage forms like binding agents, suspending agents, viscosity binders, disintegrating agents. Why not? There are many emulsifying agents. We can say thickening agent, or increasing the thixotropic characters, or controlling the thixotropic character of flow properties. And also included various compounds. Even according to me, the excipients, even containers and closures are different from this topic, but still they are also an additives or pharmaceutical aid which give the elegance. The packing also makes the patients to be more important for the particular drug to be consumed. So there are many things have to be considered when we talk about excipients. Also constitute the pharmaceutical excipients aids. We should have a discussion about it, and we should revise what you studied. You should revise what you studied in pharmaceutics. Advantages and significance of herbal excipients. Number 1, biodegradable. Number 2, biocompatible, non-toxic, biodegradable. It's very important, biodegradable. The excipients should be biodegradable and biocompatible. It should not be toxic to the patient. Economic, very important point. And safety and free from side effects and easily available. So when you see what is biodegradable, naturally occurring substances show no adverse effects on the environment or other living beings, especially human being. They are easily biodegradable. Biodegradable note are the substances which can be easily taken back into the earth when it is a waste compound, naturally without causing any harm to the environment. But here, when you are using an excipients, it should be biodegradable, means, as we told from the beginning, excipients or the additives which you are using, not the important. It doesn't have a valuable therapeutic activity. It is not involved in the active principle or active constituents which is responsible for the particular therapeutic activity. It is a supportive material for the formulation. So we need not to be harmful to our body too. So it should be biodegradable. It should be easily degrade in our body too. If we are talking about the environment here, it's a definition. It's just telling about the environment, but we are talking about something excipients which you are internally taking or externally application to the human body. So that's what very important the student should understand. Maybe I am wrong in explaining or written in the slide, but this is the main understanding for the student. When you come to biocompatible or non-toxic, It's also the most herbal excipients are carbohydrates in nature. They are compatible and non-toxic with most of the ingredients. There should be, if you have studied about any pharmacology, you might have studied about drug-drug interaction and excipients, that is substance. An interaction between the excipients also very important to study. The excipients should not react with the active ingredient or the other ingredient too. That's why we say it should be an inert substance. So economic, when you say about its advantage of economic in herbal excipients are cheaper and their cost of production is comparatively lesser when you are comparing with conventional excipients or additives which are used for formulations. Safe and free from side effects. Naturally occurring excipients are safer and without any side effects. So it's a great advantage for us to use natural excipients. Natural, easily available natural excipients are produced in most of the countries, hence easily available. And disadvantages, everything have always a merit, still means there will be a demerits. Disadvantages, some points have to be considered of herbal excipients. Microbial contamination, we should preserve it properly, otherwise when it is exposed, you can see during production of excipients, herbs are exposed to external environment, hence there are chances of microbial contaminations. Note, very important, microbiological contamination occurs due to improper processing as well as presence of excessive amount of moisture or even humid in the raw material. Number 2, a disadvantage or a demerits can be considered as biochemical variations. The variation in quality of the product from where you are bringing, different places it's grown, may occur due to variation in environmental factors because they are naturally occurring excipients. Uncontrolled rate of hydration. This occurs due to biochemical variations. As we have seen in the second point, due to biochemical variations in the environment, the difference in the quality of the material from batch to batch or batch to another, one batch to another batch. So these are some of the disadvantages. And heavy metal contamination, because most of them from natural sources from the soil. So herbs are always associated with the risk of heavy metal contamination like lead, mostly. Other heavy metals also possible because they have the contact with the earthy materials. Classification of herbal excipients, when we want to talk about herbal excipients, how they are classified. Herbal excipients can be broadly classified into the following categories based on their functions. When you see number 1, colorants, number 2, sweeteners, number 3, sweeteners binding agents, sweeteners as well as binding agents. Sorry, it is actually sweeteners, and again repeatedly it is binding agents. Let me correct it now itself so that the student will be having, when we have some mistake, it should be corrected. It is binding agents because while typing I made it together, that's why it shows like that. Anyhow, now we will see that. So when we classify, we can say number 1, colorants, number 2, sweeteners, number 3, binding agents, number 4, diluents, number 5, viscosity binders, and number 6, emulsifying agent, number 7, disintegrating agent, number 8, ointment bases, and number 9, flavoring agents, and number 10, perfumes or we have to remember all these 10 categories of classification of herbal excipients. Apart from that, you might have studied various other preparations, other needed. There are many preservatives, and you can have even stabilizers, and there are many other constituents which is supportive matters. Like if you are effervescent tablets, you need some elements should give effervescence, and when you are talking about sustained release tablets like entry coated, we need certain compounds to be encapsulated. So there are many. When you come to transdermal, when you come to other drug release drugs, when phytosomes and nanoparticles, encapsulation, all these things, when you come, you are having lots of excipients which can be used from the synthetic drug as well as naturally excipients are also available. Research are going, papers are published, students have to refer that. Number one, colorants. Example, henna. You know very well, henna we are using for every, especially marriage functions. Children and the bride and everybody were using nowadays. Henna is an important one, and even hair coloring in the North India they used to use, and even many natural coloring agent henna. And chlorophyll of a plant, caramel, amaranth, indigo. When you come to sweeteners, glycyrrhiza, honey, and stevia. Stevia is also madura chedi, but in Malayalam even they are calling as stevia. Binding agents like acacia, gelatin, tragacanth and starch. Diluents, increasing the bulkness of the active principle. If you needed it is a potent drug or for formulation purpose. Lactose, starch, mannitol, sucrose. And you can see the coloring agent. Just a picture I have taken. I will give you some examples of these pictures of the plants. Viscosity, binders like pectin, tragacanth, cellulose, guar gum, gelatin. And when we come to disintegrating agent, very important for the tablets, other drugs also, other formulations also. Example, starch, ispagul husk, carboxymethyl cellulose, all of them are natural sources. Ointment bases like lanolin, beeswax. Emulsion or emulsifying agent. We are using acacia, even agar, guar gum, methyl cellulose, tragacanth is also can be added. And when you see flavoring agent, cardamom, vanilla, lemon oil, orange oil, perfumes. There are many natural, usually natural perfumes are more, we are depending, and it's more useful and it's having less side effect when compared to synthetic products. Rose, lavender, sandalwood. So when you are coming to study of herb use as a herb as excipients, example as we told henna, when you take, it consists of dried leaves of Lavasonia inermis belonging to the family Lythraceae. Chemical constituents present in henna. Henna contains lavasone as its chief constituent. Other constituents include phenols, coumarins, flavonoids and tannins. It used as in coloring agent. Next example is amaranth. We have seen that it is an amaranth. It's a garden. Our garden plant, we used to have an ornamental plant actually. But it has a very important use in pharmaceutical additives. And you can see like coloring agent, suos, it consists of flowers of Amaranthus hypochondricus or chondriacus. Hypochondriacus and other species of Amaranthus belonging to the family Amaranthaceae. The chemical constituents contain polyphenols. We know that tannins, polyphenols, vitamins and flavonoids. I would like to tell the students, it's necessary for the students should know the structure of the compound. I have not mentioned here because the student have already come across in your 5th semester and in other subjects also. But the student should know specific chemical structures. If it is a common, then it is known as it is not a problem. If it is chemically, if they are writing some chemical specific, not the category like polyphenols, vitamins, flavonoids, no problem. But suppose if they say specifically a flavonoid name or a polyphenol name or a coumarin's name particularly present in the plant, it should be written, the structure should be written by the student. And what is the use of this dyeing agent? Amaranth is a dyeing agent. It's used as a coloring agent, used as a dyeing agent. It is used for even cloth dyeing, fabrics, and even it is used for pharmaceutical preparation. Licorice, Adi Madiram, very much useful in our traditional formulations and traditional medicine or complementary alternative medicine, Ayurveda, Siddha, and Ani. The source of licorice, it consists of dried roots and swollen of Glycyrrhiza glabra, belonging to the family Leguminosae. The chemical constituents present in licorice, it contains glyceritic acid or glycerinic acid, glyceritin, and isolicoritin. It is used as a sweetening agent, and it has many medicinal use also, not only a sweetening agent. Stevia, not stilva, I told previously, it was stevia or stevia. Stevia, in Malayalam also they used to say stevia sometimes, but its name is Madura Chedi. In Tamil it is called Madura Kirai. Madura, not Madurai. Madura Chedi, Madura. Madura means sweet, Madura Kirai. So when you see sources, it consists of the plant of stevia, Rebaudiana, belonging to the family Compositae. Chemical constituent, it consists of stevioside, a glycoside, stevioside, rebidioside, steviol. At least these all are specific constituents, not a general constituents. We did not say glycoside, alkaloid, flavonoids, tannins, or polyphenols. Simply we did not say. We have given the name particularly. So at least a structure of this compound have to be specified by the student when they are writing an example like stevia as a sweetening agent, as a natural sweetening agent. Interesting to know, used as a sweetening agent. When you come to acacia, well known, you know that it is an unorganized drug. It's an exudate. It's derived from the plant Acacia tree. It's a gummy exudation obtained from the stems and the branches of Acacia arabica, belonging to the family Leguminosae. The chemical constituent, it contains sugars like arabinose, galactose, ravenose, and glycerinic acid. Highly concentrated, highly capacity with the carbohydrate. Acacia is used as binding agent, suspending agent, increases the viscosity, emulsifying agent, reduces the surface tension between the two immiscible liquids, and viscosity binders. So interesting application of acacia. Gelatin, we know that gelatin, hard gelatin capsules, soft gelatin capsules, which we are using commonly for various drugs in our pharmaceutical industry. Even in herbal formulations, to make it elegant and to make it easy for the consuming for the patients. We have different colors and appearance of soft gelatin capsules and hard gelatin capsules. The source usually it's a protein obtained by partial hydrolysis of animal connective tissues like bones, skin, tendons and ligaments obtained from animals. Chemical constituents gelatin chemically contain amino acids like glycine, alanine, glutamic acid, proline, arginine, aspartic acid, leucine, isoleucine. Commonly, it has all the aliphatic amino acid or essential amino acids. So interesting the uses of gelatin. We know that it is used as a binding agent, thickening agent, emulsifying agent, and it is for manufacturing of capsules of soft gelatin capsules and hard gelatin capsules as shown in the picture. Tragagan, an interesting natural product. It's also an exudate. It's also an unorganized drug derived from the plant. It's also a gummy exudate obtained from, it's a dried gummy exudation obtained from the stems of branches of Astragalus gummifer. Astragalus gummifer belonging to the family Leguminosae. The chemical constituents, it contains gums, tragaganthine, basorin. Uses, what's the use? It is used as binding agent and viscosity binders. So we should know, you should know about what's the chemical structure of tragaganthine, what's the chemical structure of basorin is an interesting for you to specify. It's an interesting product, tragagan. Lactose, we know that it is an disaccharides and it is used mostly in as a filler. It's a natural disaccharide obtained from milk. Chemical constituents, it contains sugar galactose and glucose. It is used as a diluent in tablets and capsules. Now coming to mannitol, an interesting product and a sweetening agent. And even it is in diluent, it can be used as in bulk, increasing the bulk of the potent ingredients, and it can be used for various liquid orals preparations. The source, it is a saccharin. It is a saccharin. It is a carbohydrate, saccharide, exudation from the stems of Fraxinus ornus, belonging to the family Oleaceae. You see the plant flower, it's available in our place also, mania, ash, but very rare. Fraxinus ornus plant. You see the picture of the exudate which is coming from the stem, which is the side branch, used as a sweetening agent and as a diluent. Interesting to see that how slight creamish color, white after purifying. Guar gum is also a powder which is obtained from the seeds of this vegetable, which we are using very common and usually we used to say that it is very good for diabetic patient. Yes, it's true. It has a very good content to stimulate the pancreas or the islets of pancreas to secrete insulin, and it can be used for even insufficient insulin in this, that is type 2 type of diabetic mellitus. So the plant is Cymopus tetragonolubus. So the species is tetragonolubus, and it is in Tamil, it is Kodavarai, that is Kodavarai, they used to say Kodavarai, and in Malayalam you can say Kodamara, and that is you can say Kodamare, or you can say in locally they used to say Chinvarikai. and it is dried, and the seeds you can see very clearly, but we used to use in our vegetable preparation, especially in avial or thuran, they used to prepare, and guar gum. But this is we prepared as a powder from the seeds. The source is the ground endosperm of the seeds of Cymoplis or Cymoplis tetragonolobus, belonging to the family Leguminosae. The chemical constituent present contains 85% of guaran, galactose and maltose. It is used as a binding agent, disintegrating agent, suspending agent and emulsifying agent. So interesting application of guar gum. Starch, we know there are many types of starch obtained. It consists of polysaccharide granules obtained from the grains of maize, rice, wheat, potato, tapioca. There are many other sources are also available. And chemical constituents, it consists water soluble amylase and water insoluble amylopectin. We know the structure, we have studied in detail about this polysaccharide, which swells and is responsible for gelatinizing property of starch, which I am talking about amylopectin part of that is insoluble amylopectin, which has the gelatinizing part of the starch. It is used as a binding agent and as well as disintegrating agent and diluent. It has 3 varieties of uses in one formulation like tablets. So we should know how it acts as a binding agent, how it acts and where it acts as a disintegrating agent, and how it acts as a diluent. The student should know very clearly, and he should have the idea about the definitions of the various excipients and the examples when you study, so that you can elaborate your answers when they ask even 5 mark or 10 mark. Detail about the excipients can be explained by the students. So as we have seen, that is, it can be obtained from even potato, maize, and even from the wheat, rice. Sandalwood oil, the next example, the source, it is an volatile oil obtained from the steam distillation of hot wood of Xanthalum album, belonging to the family Sandalaceae. Chemical constituents present in the sandalwood, it consists of sesquiterpene alcohol, namely alpha and beta xanthalol. Uses, it is used in perfumery industry. We know that very well, interesting tree of sandalwood. Flower, rose oil, rose flower is very common as an ornamental plant and used for various purposes. Different varieties of roses are available, we know that. Sources, it is volatile oil distilled or by infrared technique like French. The new technique can also be used for fresh flowers of Rosa gallica and other rose species belonging to the family Rosaceae. Chemical constituent present in the rose flower, rose oil consists of linalool, a terpene, monoterpene, nerol, citronellol and geraniol. Uses, it is used as a perfumery industry, or it is used as a perfumes and flavoring agents in various formulations. When we come to cardamom oil, we know that plant cardamom very much cultivated in Idiki districts and other hilly areas. It needs more care, but it gives more price for the cultivator. Sources, it is volatile oil distilled from the seeds of Eleutheria cardamomum, belonging to the family Gingiberaceae. Chemical constituent, it consists of cineol, alpha terpenyl acetate, terpeneol, borneol and sabinin. Uses, used as a flavoring agent. When we come to the next one, orange oil. orange oil. It is volatile oil obtained by the expression from the fresh peels of the ripe fruits of citrus Lemonia belonging to the family Rutaceae. Chemical constituents present in rose oil contains limonene, citral and citronella. It is used as a flavoring agent. Now we are coming to the herbal formulations, which I am discussing very briefly, but the student have to use application what you studied in pharmaceutics. The procedures have to be added in every formulations. Herbal or herbs have been used as a wide variety of dosage forms. Large number of herbal formulations containing the plants or their extracts with proven medicinal activity are being used for herbal formulation. With the advantage of pharmaceutical technology, modern dosage forms have been evolved. The herbal formulation dosage form can be broadly classified. One is traditional dosage form, modern herbal dosage form, novel drug delivery system, novel dosage form. Herbal formulation may contain a single herb or combination of different herbs. So traditional dosage forms, these are derived from various traditional system of medicine like Ayurveda, Unani, Siddha, homeopathy, etc. It may be pills, example, powder, pills like tablets, mold tablets or made tablets or even compressed tablets is available, semi-fluid extracts, pellets, tinctures, etc. Modern herbal formulation, when you see these formulations are developed by modern technology process, modern herbal formulations offer small dosage sizes, single dosage sizes. They are user friendly, they are elegant, they are attractive, they are convenient for the patient to have good absorption also. Characteristic like tablets, capsules, syrup, solutions, suppositories, injections. And when we come to novel drug delivery system, there are many. Like in cosmetics, you have come across serum, you have come across transdermal applications, and with an advantage of different, and even for the formulation when you see vaccines, different formulations, and also phytosomes, these all, and also nanoparticles, enricoated, these all, and even encapsulations, micro-encapsulation, this all comes under the novel drug delivery system. With an advancement of the different scientific techniques, the preparation of formulas or formulations, novel dosage forms are being developed to overcome the limitations of conventional dosage forms such as tablets, syrups and solutions. Many novel dosage forms have been developed successfully which have offered better acceptance by the health system. A delivery system was very important. And even the less dosage form, it can have the same therapeutic effect which you are given in the normal dosage form. A few novel dosage forms are available in the market as transdermal implant, nasal systems, microcapsules, microspheres, liposomes, phytosomes, etc. So herbal formulations simply we can classify like syrups, tablets, ointments, creams and even capsules and mixtures. Novel dosage forms we are going to have a discussion about even how to prepare phytosomes, liposomes, nanoparticles and the microspheres. So just a brief idea about this I am going to discuss with you. And you can see that herbal syrups, we know herbal syrups which are mostly available in the market and have a high demand. These are preparation formulated by incorporating sugar solution, as we know that how to prepare a syrup. with plant extracts such as infusion, decoction, juices, fermented products like arista, aswa, or simple solution can be added. Honey or unrefined sugar is used to prepare syrups as they act as good preservative. Herbal syrups are made with equal proportions of herbal extracts with honey or sugar solution of known concentration. Various flavors like orange, raspberry, mentha can also be added to mask the bitter taste as well as improve the palatability of the formulation. Herbal tablets, the student should have a basic idea, and to be come here I am not going to explain about the tablet formulations, how you can make by direct compression method, wet granulation method, dry granulation method, mold method. These all you have to refer in your pharmaceutics what you studied, and it can be applied only incorporating the herbal materials as an active principle in that particular. Other all things you can have even add the excipients. You can substitute the excipients what is available as a herbal formulations. So these all are some of the herbal tablets which I have just given a picture. These are solid or a unit dosage form of powdered herbs. Extracts, herbal extracts, or their constituents isolated can be prepared by molding or by compression. Just I have given only but there are many procedures which you studied. Pages together you can write. In addition to the active ingredient, these constituents diluents like binding agents which are provided strength to withstand the normal handling while transporting and storage. So we have many evaluation in tablets, in process tablets like friability and hardness testing. These are all the things which is we are telling you in a one line. Coloring agents to improve the appearance, sweetening and flavoring agent to mask the bitter taste. Disintegrating agents to facilitate the breakdown of the absorption, breakdown of the formulation, breakdown of the tablet to give the pharmacokinetics of absorption in the gastrointestinal tract are added. And herbal ointments, you have ointment base which we come across in the preparation of ointments. The first experiment which you given in the pharmaceutics might be prepared in sulfur ointment, I hope. How to use the spatula, how to prepare a pharmaceutical ointment base. So here also we have to go for herbal formulation. These are semi-solid dosage forms meant for external application to the skin or the mucous membrane. Ointment perform softening, emollient and protective action. So we have many waxes of beeswax, paraffin wax are used as a base, ointment base of carrier. Even cocoa butter. There are many other natural bases are available. Various active herbal ingredients in the form of powder, dried extract can be incorporated in the base and provide therapeutic benefits. Herbal creams, you know that difference between cream and ointment like suspension and emulsion. Creams are oil in water, emulsion you can see water in oil type is mostly. So when you see types are viscous, semi-solid preparation which may be oil in water type, aqueous creams, or water in oil type, oil greasy creams. So when you see herbal mixtures, interesting to see the pictures and the formulas which is available in the market. Mixtures means actually we may think that is mixture only the solution. No. Mixing of components, mixing of constituents, combination of many constituents usually seen in herbal preparation. These are preparation containing combination of 2 or more herbal ingredients which are formulated in various formulations like tablet, capsule, ointments, creams and etc. which I mentioned that is maybe liquid oral preparations as we have studied in mixtures also. These mixtures or combination of herbal ingredients may have more than one activity or one active ingredient can be used to treat multiple ailments, multiple diseases, or they may produce additive or synergetic effect when additive, this you know that additive reaction, synergetic reaction. You might have seen, you might have studied about this in your pharmacology, basic pharmacology initially. So two are molecules which they increase added therapeutic activity, synergetic multiplication of synergetic activity, how the alcohol and the phenobarbital makes the synergetic effect of single disease. So this is also important point which I want to explain. Now we are coming to the novel dosage form where we have to discuss about phytosomes, how to prepare, and the liposomes definitions, what is there, and nanoparticles and microspheres which you are coming across in your biopharmaceutics. But still I have given some of the brief explanation for the students to have an idea. Suppose if they ask a 5-mark question or a 10-mark question about novel dosage forms, explain about phytosomes, liposomes, nanoparticles, microspheres which can be prepared in herbal formulation. If they ask, the student have to have them easy to write about that if you listen to this class. And if you listen, please kindly make it a notes and you will succeed. It's an interesting for even an 8th sem students, even for the other students also. Phytosomes, preparation of phytosomes we are going to see now, and we are going to see how to prepare by means of anti-solvent precipitation technique, how they will prepare the phytosomes, and rotary evaporation techniques can be applied for preparation of phytosomes, and the solvent evaporation method. So 3 method of preparation of phytosomes we are going to see, so that it will be easy for you to know about how can you prepare a novel dosage form, how can you prepare a phytosomes in herbal formulation, can be easily explained, and what type of chemicals can be added. Novel drug delivery system, NDDS, novel dosage forms. And when you want to see, this is phytosomes, you see the picture, how the puts and positive ion charged, that is the means the host, that is just to mention that curcumin, the isolated compound from the curcuma longa. And you can see that is phospholipids which has a tail and a head. So just to show you how the tail works, how the body works, just to show them. The novel dosage forms usually contain pure isolated compound, herbal, like as shown in the picture, curcumin, nanoparticles, and the colloidal system are particle size of about 1 to 1000 nanometer or micron. So very 1 to even 1 micron substance. And phytosomes, what is phytosomes? The concept of phytosomes, phyto, the plant chemicals, plant constituents, specific active principle. Phytosomes are another breakthrough for the development of herbal drug technology. It's an interesting technology for herbal drug technology. Phytosomes contain active herbal ingredients surrounded, as shown in the picture, surrounded and bounded by phospholipids. They are produced by bonding, as shown, bonding with, the active principle will be bonded with phosphatidyl, or you can say phosphatidyl choline, phosphatidyl choline, or phosphatized phosphatidyl choline with the ingredients, and thereby producing a complex. The phospholipid structure has a water-soluble choline. Phospholipid structure has a water-soluble choline. This has to be remembered by the student. Phospholipids, choline, and phosphatidyl. These things, if they remember, you can write the answer very clearly with the picture. Head, that choline is present in the head, and the fat-soluble phosphatidyl is present in the body and the tail. Very simple. Choline is water-soluble, which is present in the head, and fat-soluble is present in the body and the tail, that is phosphatidyl portion. So the choline head water-soluble binds with the active compounds, herbal ingredients, water soluble, because we need water soluble components to be distributed, or you have to need to go for dilution solution in our body fluid. So the choline head water soluble binds the active components, herbal ingredients. If you are taking curcumin, curcumin will be binded with the choline, while the phosphatidyle or phosphatidyl portion, fat soluble, comprise of the body, and the tail also envelops the choline bounded material. Tail also, you see, only the body, tail. So it has a multi appearance of solubility with the water soluble component. Vesicle are produced whereby valuable component of herbal are protected. Phytosomes enhance the absorption and improve the bioavailability of the herbal ingredients by enhancing their delivery and the tissue, thereby another important point is lowering its doses. You are lowering the component to enter into our body. With a low dose, having the good therapeutic activity, a very great advantage and very great scientific proved, and to identify to have the phytosomes lowering its dose and reducing the side effect. They are also protect the active, they will also have the protective stability given to the herbal ingredients from the destruction of the digestive juices and gut bacteria. Several studies have been shown that the body uses phytosomes molecules more effectively than the non-phytosome molecules. Preparation of phytosomes, we have three types we are going to discuss. Phytosomes are generally prepared by adding accurate amount of phospholipids, soya lecithin, with herbal extracts in a aprotic solvent. Soya lecithin contain a main constituent of phosphatidyl choline, which is having a double function. Phosphatidyl part lipophilic nature and choline part is hydrophilic nature, we know very well. The choline part is attached with the hydrophilic main active constituents, whereas the phosphatidyl part is lipid soluble component or lipid soluble part attached with the choline bound complex. It results the formation of lipid complex with better solubility and bioavailability. It can be prepared by the method number 1. We are going to discuss about anti-solvent precipitation technique. The specific amount of the drug soya lecithin or soya lecithin were taken in 100 ml of round bottom flask and fixed with a reflex with 20 ml of dichloromethane at a temperature not exceeding 60 degrees centigrade for 2 hours. Reflex. You know that reflex, how they fix in the round bottom flask. The mixture is concentrated, heated 5 to 10 ml. You added 20, it is made into very minimum 5 to 10 ml. Hexane 20 ml is now added carefully with continuous stirring. To get and precipitate, you will get precipitate when you add hexane, a very light non-polar solvent which was filtered and collected and stored in a vacuum desiccator overnight. So you got the precipitate. The dried precipitate is crushed in mortar and passed through the sieve of, it should be fine in an 100 mesh, 33 mesh, 80 mesh. 100 mesh means very, very fine, can be sieved. The holes of the sieve will be very small. 100 holes will be there in 1 square millimeter. You know very well about the sieve mesh. Powdered complex was placed in an amber colored bottle and stored in a room temperature. So you see, this is the procedure what you studied, and a picture which is shown here. This is prepared by me for you to have an understanding. A drug and soya lecithin can be added with 20 ml of dichloromethane. You have to add drug in the initial stage itself. Drug and the soya lecithin with 20 ml of dichloromethane in 60 degree in a reflex. You have to make continuous stirring precipitate, and you are adding with concentrated, and you are after concentrating you are adding hexane of 20 ml. So stored in a vacuum. After getting the precipitate, it is stored in a vacuum for overnight, and then the precipitate which you taken from the desiccator is crushed and passed through a mesh of 100 mesh sieve, and then it can be stored in an amber colored bottle because it is light sensitive. So anti-solvent precipitate technique can be used for preparing phytosomes. Number 2, rotary evaporating technique. Here the specific amount of the drug plus soya lecithin, same soya lecithin, remember, were dissolved in 30 ml of tetrahydrofuran. We are adding with tetrahydrofuran in a rotary round bottom flask, followed by stirring with 3 hours at temperature not exceeding 40 degrees centigrade. The film of the sample was obtained. A film of the sample is obtained, to which now you are adding N-hexane, was added and continuous stirring using a magnetic stirrer. So this is rotary evaporating technique. The precipitate obtained is connected and placed in an amber colored bottle and stored in a room temperature. Here you can see there is a stirrer. Magnetic stirrer will be kept in a round bottom flask. A drug and soya lecithin were dissolved in 30 ml of tetrahydrofuran. You have to remember the tetrahydrofuran. Stir for 3 hours at a temperature not exceeding 40 degrees centigrade. That's why we have kept a mandel also. And a film of sample plus N-hexane continuously stirring. You should stir with a magnetic stirrer. The precipitate is stored in an amber colored bottle during room temperature. Solvent evaporation method is an interesting method. You can see the specific amount of drug and the soya lecithin were added in 100 ml of round bottom flask, reflex as we did in the first one. 20 ml of acetone here. You have to remember the chemicals which you are using in different forms. Acetone at a temperature of 50 to 60 degrees centigrade for 2 hours. The mixture is concentrated to 5 to 10 ml to obtain a precipitate which can be filtered and collected. The dried precipitate complex was placed in an amber colored bottle, glass bottle, and stored in a room temperature. You can see the picture how we did with a drug of soya lecithin were taken in a 100 ml round bottom flask with a reflex. You can see the picture of reflex which I have kept the condenser, which will not make the solvent to evaporate and to get outside. It will be condensed. 20 ml of acetone is added at a temperature of 50 to 60 degrees centigrade for 2 hours. And then the concentrated, you are making it to concentrate for 5 to 10 ml, reducing the amount of ml quantity obtained precipitate. And this precipitate is again filtered and collected, and the dried precipitate complex was placed in an amber colored bottle as shown in the picture to provide a shelter from the direct light sensitivity. So this is about the next one. First, phytosomes. What are phytosomes? We have talked about the phospholipids and phosphatization of the choline. We have discussed about the phytosomes and how to prepare the 3 techniques. Interesting to know about them with the picture. And you can see now the second one, liposomes or liposomes. Liposomes, you can see there is an hydrophilic head, hydrophilic tail and an aqueous interior, the structure of unicellular liposomes with a lipophilic molecule. So these are prepared by incorporating the active ingredients side by the microscopic double layered membranes which are made up of lipids known as phospholipid vesicles. These vesicles are suspended in an aqueous solvent uniformly. Drugs incorporated in the liposomes can be delivered to the desired site in desired concentration. This novel drug delivery system is especially targeted to liposomes delivery of drug in cancer chemotherapy, arthritis, hemophilia, and the disease of immune system. You can see this aqueous interior face and the aqueous external face with the structure of this which I have shown there. It's an interesting to know about them. A unicellular liposomes is a spherical chamber vesicle bounded by a single bilayer of an amphiphilic liquid of a mixture of such lipids containing aqueous solution inside the chamber. The unilaminar liposomes are used to study biological system and to mimic the same cell membrane. This is about nanoparticles, which you might have studied in your pharmaceutics very more than this. What I am saying is just in brief definition. These are colloidal particles of the submicron size which acts as a carrier for drug molecules. Maybe a herbal molecules you can incorporate. These are used to target sites in the treatment of cancer, disease of reticuloendothelial system, and enzyme replacement therapy in liver. And a microsphere cross section of the cell, you can see the polymer 1, polymer 2 dispersed drugs can be made in microspheres. When you talk about microspheres, these are small solid particles carrier containing the dispersed drug particles either in solution or crystalline form. The drug can be made. The microspheres are used as carriers for drugs and therapeutic agents, especially in cancer treatment and hormonal disorders. So anyhow, we have given an explanation about this chapter briefly. 4th unit. Kindly students concentrate in this herbal cosmetics and formulations. They are asking many questions starting from the first lecture and second lecture. Please listen and write down very clear assignment and submit to my office. Again, I inform you the students, kindly subscribe my channel. Thank you.